Well, welcome to the final session of Bioassays 2020. I hope everyone's had a great experience with this virtual. It's unfortunate we weren't all face-to-face, but I think the virtual, from what I've seen, has gone off quite well. And I thank you for participating, and I hope you've gotten out of this as much as I have, which has been quite a bit. It's been quite enjoyable. It is now my pleasure to introduce to you Steve Kozlowski. Probably Steve goes without needing to be introduced, but for those of you that have been hiding under a rock lately, I guess I'll go ahead and do it. Steve is the Director of Office of Biological Products, OBP, Office of Pharmaceutical Quality, and the Center of Drug Evaluation and Research, better known as CDER, at the FDA. The OBP is responsible for quality review of monoclonal antibodies and most therapeutic proteins. They also provide immunological response of therapeutic proteins and performs missions related to research. I hope you will enjoy Steve's talk as much as I do. Steve will talk for approximately 40 minutes, and we're leaving 5 minutes on the back end for questions. So please use the chat room for your questions, and we'll get them to Steve. And with that, Steve, the monitor is yours.
Thank you very much. So I gave a talk very similar to the talk that I'm giving now at the CE Pharma meeting, and it was also the last talk of the meeting. So aspects of giving the last talk of the meeting are many people come up to you right before and say, sorry, I have to leave because I have a flight to catch. Even the people who are there have their luggage sort of in their hand and a coat folded over it and are kind of heading out the door. So I kind of wonder whether in the virtual environment, is that a little more favorable for being the last talk in a session, as at least one can't claim that one is heading out because of a flight issue. Another aspect of virtual meetings is that COVID-19 has made us suddenly, figure out how to do them. But the technology has always been there. I know I've participated virtually in meetings that were not virtual in the past for reasons, but somehow we've had to take the pre-existing framework and platforms and convert them rapidly to be able to meet this unexpected but very critical need. So one of the things that I will talk about today is frameworks for protein analytics. And I would pose that just like having frameworks in place allowed us to go virtual when we needed to, having the right framework and structures for how we think about protein analytics, including bioassays, will really position us to deal with challenges as they come up and to really accelerate pharmaceutical development. So that's basically the main theme of what I'm going to talk about. But many times when industry presents, if you're from a company, you'll have a set of slides that are kind of your company slides. You have to put out the message before you talk about details. So the FDA is following that example. And so I will start with a few slides on the FDA perspective on pharmaceutical quality that I think tends to be a good way of starting conversations about quality. So obviously, quality is a term that gets used a lot, and quality in the context of other products, whether a cell phone or a car, is really meeting user expectations, not meeting a regulatory standard and not meeting CGMP, although obviously those are standards that are important, but quality really means meeting the expectations of the user, and drugs are no different. And so, patients, really our ultimate stakeholders, expect safe and effective medicine with every single dose they take. And pharmaceutical quality is assuring every dose is safe and effective, free of contamination and defects. And I think, quality is often separated from safety and efficacy, but the reality is quality is safety and efficacy, but at the individual dose level. And it's really critical because it gives patients confidence every time they take their medicines. So with that brief introduction, this higher arching goal of quality, obviously that needs to be translated into a variety of other things to ensure that the patient gets what they expect in each dose. And for biologics, quality has evolved with analytics. That historically, biologic products were defined by the process, and there were even times when there was a small, probably meaningless clinical trial associated with manufacturing changes simply because there was no confidence in the ability of analytics to discern whether there were important changes with that manufacturing change. And so analytics have let us create an alternative to clinical data for every manufacturing change, and that is quality attributes. And quality attributes play a role directly in control strategies such as specifications and other roles, in comparability, enabling manufacturing changes. Key for the development of the biotech industry has the ability to scale up without having to do new clinical trials. And obviously, good analytics can lower overall costs and increase access through biosimilars. And it also helps us understand variability and mitigate risks in a broad sense related to adverse events and shortages. So analytics are extremely important. There are many analytical tools that evaluate proteins, tools that evaluate primary structure, tools that evaluate higher order structure, ranging from things like disulfide bonding to sophisticated spectrometry to directly measuring structure through things like NMR. Looking at the heterogeneity of the products we regulate by various characteristics, whether size, charge, or hydrophobicity, glycosylation of proteins, and many other things, pegylation, and obviously bioactivity. The key part of this meeting is a very, very important part of analytical tools for protein, a surrogate for higher order structure, but much more than that because it's the true test of a product activity closest to patient needs. So there are many, many tools, and they measure many, many attributes. And there are very similar tools across many of the attributes that are mentioned, at least general mechanisms for tools. And so how do you organize all this? What's the most useful way of thinking about this? And clearly, attributes, there are many more of them even than the many methods, but attributes are the bottom line because they are what correlates with what the patient experiences. So ideally, we would want to organize all this based on attributes. So what are examples of how this gets organized? So in a regulatory world, we have regulatory categories for attributes. And, lot release might be defined appearance and description, identity, purity and impurities, potency, and quantity. But in a scientific sense, those are really not that helpful, right? Obviously, appearance is macroscopic physical attributes, but identity and purity and impurities overlap. The same assays and the same attributes can fit into both of those categories. Similarly, process-related impurities are probably mostly in the impurity category, but again, product-related impurities really can fall into many places. Potency, protein function, bioassay binding, so that generally lives in that category. But all these things, independent of being related to specifications, may be related to deep characterization, which could define critical quality attributes, ultimately comparability, analytical similarity. The attributes might be looked at in different ways over those things. So again, what is the best way to organize this information in a framework? So I'm going to throw out some categories. We know in this, science, there'll be a million views of this, some of them based on tradition, your education, different views about what's important, but clearly size in some manner, charge in some manner, whether polarity is a subset of charge or you treat it separately, hydrophobicity, higher order structure, primary sequence, modifications to the protein sequence, and potentially treating some modifications differently that are, polymers. Glycosylation probably being the most common example of that, but others like pegylation, things where we actually, artificially change the proteins. So how could all of this be organized in a useful way? And so that's really the question I'm asking with this part of the talk. So if you think about protein attributes, what are dimensions that you look at? So one dimension is resolution. You can have a measurement which gives you an average value for each molecule. You can have a metric which defines atomic level structure of attributes. And often we're in the middle of those with some sort of pattern associated with attributes like spectrometry. So resolution is one aspect of the attribute you're looking at. Are you looking at for the average molecular weight of all the molecules in the vial, or are you looking at something much more detailed than that? Similarly, sensitivity is another general category of attribute measurements. Are you looking at the predominant form of something, or are you looking down to extremely low variance? And then ultimately, from the point of view of regulatory perspective, is what are the relevance of those attributes, right? It may be very, very rare variants are extremely interesting from an academic perspective, but if they're not relevant to the patient, then they may not be important in terms of how you control and manufacture a product. Now, these three dimensions apply to all measurements. There is one regulatory category of measurement that's a little bit of an exception, and that's identity, which is the ability to separate your product from other products. So that may not depend on relevance as much as other attributes, and it's often in the category of high sensitivity and high resolution because by definition, it needs to be distinguishing. And so you need to have enough granularity to use it to distinguish. But for most other things we care about, they fall somewhere into this three-dimensional space. So, and I've given this talk to people who are more involved in aspects of protein analytics other than bioassays. I'm going to give some examples, and I'm going to have artificial guesses by me of resolution, sensitivity, and relevance. And those are for illustrative purposes. No real products were injured or maligned in demonstrating this, and that no actual reputations should be impacted in terms of particular methods. So whatever you do, bioassays, for instance, this crowd obviously has people with the best resolution and sensitivity, and examples don't comment on that. So taking this construct of resolution versus sensitivity, and this is very similar in some sense to talking about top-down and bottom-up in the mass spectrometry world. Are you really looking at a measurement of a 15 nanometer entity, like maybe a whole antibody, or are you looking at, a sub-nanometer measurement, such as an amino acid or below? And let's take charge. So some potential analytics for charge, capillary IEF, or breaking an, product into subunits, and then looking at the isoelectric focusing. And so two types of assays. If you're not breaking down the molecule, then it's at a single value. You're getting the average PI for that entire molecule. If you break it down into subunits, you're getting a little more resolution. You could also have a peptide map where you do a digest and you run it over a charge-based column. And you can also use multi-attribute methods like mass spec and use sequence data to define charge. So a variety of ways of looking at charge at a variety of levels. And that third dimension would play in this way. If your product, the isoelectric point is some way correlated with bioavailability, it's a sub-Q injection and there's some known impact of PI of the whole molecule, then that actually becomes important to measure at that level of resolution. If, on the other hand, you're interested in a very specific deamidation that impacts potency and not overall charge, then a multi-attribute mass spec method might be the right tool for that problem. And again, on the sensitivity axis, which these, I'm assuming they're all between 1 and 10% overall sensitivity to variance, if you care about a lower level of a variant, then you want a method that moves more into that space. Another example, higher order structure. So crystal structure, right? The gold standard, maybe that's being challenged in some ways, but certainly historically the gold standard for structure. It's atomic level, right? It's very, very high resolution. On the other hand, it's only the predominant form. It's usually the form that's able to crystallize, and it really doesn't tell you much about variance. 2D NMR may be able to give you more ability to look at variance. Spectrometry method might give you some, idea of some, say, secondary structure folding, but is really not going to give you, very granular levels. Cross-linking, three-dimensional structure, breaking it up, and then using mass spec can give you maybe a little more granular sense about how the molecule folds, hydrogen-deuterium exchange. So again, in this play of methods for detecting higher order structure, what's the third dimension? So if misfolded protein has decreased potency and a spectrometry method is sufficient to detect that, then that may be what you need. On the other hand, if there's a particular misfolding that you know of that has increased immunogenicity, then you might need a more granular method to be able to look at that. And so the concept is to create a framework like this and have an attribute tree that is organized by this framework that both goes into different levels of resolution and that considers interactions, because this tree won't be simple, because things at the amino acid level are going to affect hydrophobicity and maybe charge at the same time and size. So really creating a map for this, and obviously considering sensitivity in that map. So what is the value of having something like this? So I have certainly heard in communications from people in industry, companies have their own frameworks for this, because it's very, very important for them to track attributes and to have knowledge management. But as far as I know, there is no public framework that uses the same organization and maybe even more importantly, the same nomenclature. I will certainly say even afucosylation of monoclonal antibodies, how that's described, what structures it includes, varies from company to company based on their own methods. So if you really wanted to be able to look across that, there is no easy way without a lot of manual labor to understand that. So what would such a public framework give us? So it could impact both public and private data. So clearly product specs are private commercial data, and they could be organized this way in interactions between regulatory agencies and industry and only shared at that level. Similarly, any shared proprietary analytics for a control strategy, for manufacturing process, that framework would just be a way of communicating between regulators and industry in private. Analytical methods, some of them may be proprietary, some of them may be public. Both public and private methods could be organized in how they fit in the framework. And then all public data, published data, public databases, maybe even wikis that people create on analytics, if they have a shared framework, then all that data can be more easily accessed and used and understood. So if we really wanted to do this, and again, I put in a little plug for the FDA. So you may have heard of something called CASA, knowledge aided, and structured applications. So basically it's an idea for knowledge aided assessments and structured applications, a way of organizing our reviews and hopefully ultimately getting structured information to match that to really improve our efficiency in terms of reviewing applications for industry. Having a system like this might be sort of a core engine of that type of structured assessment. So what exists, right? You would think that there are many standards. What standards exist that fit this? So ISO has a standard for identifying medicinal products, and specified substance is one piece of that standard. So when it comes to proteins, the level 0 or group 0 has very high level information, amino acid sequence. It won't tell you the exact glycosylation, but it may indicate it. Is it mammalian? Is it yeast-like? Some general characteristics of glycosylation and potentially glycosylation sites, sometimes disulfide bonds and other modifications, but certainly not the granularity that is needed to describe products in a very meaningful way from a regulatory purpose. There is a group 1, which I don't think that's really been defined that well yet, which is the next layer under. That may be an opportunity for a shared framework. Manufacturer is group 2. Again, that may be in the private domain. There are shared standards when they're available, which might be WHO or some other shared standard. And Manufacturer specifications would be private, but they could be organized in this same way. So this is a standard. Group 1 is not really complete. How would this standard be stored? So there is the Genus Project Global Ingredient Archival System. So that's the broad system, and it relates to identifying unique codes for each ingredient, known as a uni, which many of you may be familiar with as an organizing way of substances. And there's a database, so there's Genus is sort of like the overall program. There's a database, the Global Substance Registration System called GSRS. That system is meant to be ISO compliant. It's developed by NIH, but closely works with FDA informatics. And that's where unis are assigned. So if you have a product you're developing and there is a uni associated with it, it is that system that classified that ingredient or that active ingredient with a uni. And so this system exists, and I want to give you an example of some public information on that system. So, siltuximab is an approved antibody. This was on the public side of this data set. So it gives you the sequences by subunit. It gives you disulfide links. It mentions the glycosylation sites. And it has some general information like an estimated molecular weight. Very much like this group 0 ISO standard amino acid sequence type of glycosylation and some level of modification. So if there was a way of taking group 1 and making this useful as a shared interface for protein analytical information, that would certainly fit into existing systems. It doesn't mean that it has to, but this would have the advantage of being part of an accepted standard and part of an IT system that's already being developed around it. So I want to talk a little more about what this map could look like. And again, this is where you'd get into an awful lot of debates. So product attributes include process impurities, excipients, and I want to focus on protein attributes, a particular subset of product attributes. And there are functional attributes, very, very relevant to this meeting. And those may involve defining what the target is, whether you're then measuring biological activity of engaging that target or whether you're measuring binding. And for structure, there's primary structure and higher order structure, I think, which there will be little debate over. And then do you treat glycoforms and other polymers as a separate category or not? And then what general characteristics do you consider categories? Charge, hydrophobicity, again, would polarity be separate? If you have a high-level map like this, some of the information, I'm just throwing out things that would be in GSRS exist, like target might be included, disulfide links, glycosylation, but only a very limited level of that. So taking structure, what would a deeper dive in structure look like in those categories? Using this top-down, bottom-up approach for resolution, charge, right? Isoelectric point would be the whole molecule, and then molecular charge at the amino acid level would be the more high-resolution method. Similarly, overall mass to mass impacted by specific amino acid modifications. And these interconnect. Amino acid modifications may affect many, many branches of this tree. And similarly, you could have high and low level polymer composition. What is it? Is it a sugar? Is it polyethylene glycol? What is the polymer? And then mass, and then just like you can look at peptides, you can look at oligosaccharides or polymer fragments, finally down to the particular molecular structure of the polymer and the linkage chemistry. Similarly, you can have an indirect correlate of higher order structure. You can have something that roughly correlates with some level of structure, and you can go down to the atomic level. And hydrophobicity also, at what level are you looking at? So there's a way of organizing, I think, most of what we would look at. For function, again, in a top-down manner, is your bioassay a whole animal, right? That's very, very rare nowadays, but we still have some products which intermittently use whole animal bioassays. A whole tissue or organ, we're not necessarily there yet in most cases, but, with organs on a chip, there may be bioassays that live at that level. Cell culture, and then clone cell lines, and then a molecular target on the line you're looking at, and finally a variant of that target, whether a mutation, like we look at variants for FC receptors in assessing things. So there's a way of framing the target in this system. And then biological activity. Is it something very high level, survival, proliferation, or is it very, very, granular, such as phosphorylation of a particular signaling molecule in a signaling pathway for a receptor? And again, these things interconnect in different places. And then binding, are you looking at a single parameter of binding, an equilibrium constant, or are you looking at much more sophisticated binding kinetics, and how, again, these things might relate together? So I've spent the first part, and so I don't know if my clock started at 1:30 or 1:35 when I started, but my goal is to be about 40 minutes of talking. So I'm going to aim to be done in about 15 minutes and have 5 minutes of questions. If that's too long, somebody let me know. So the next thing I want to talk about is this red axis of relevance, because this is critical, right? Again, even if we do have a great way of organizing our attributes, we want to focus on the ones that really matter from a regulatory point of view. Because our job ultimately, like the first few slides I showed at the beginning, is what matters to the patient for each dose of product they take. So how do we establish relevance? And so we generally do it by risk assessment. So this is from, a very old example, AMAB, which talked about risk assessment for particular quality attributes. Generally, some combination of prior knowledge, clinical studies maybe for a related product if you don't have it for that study, for that product, animal data, in vitro data, somehow is all integrated into a risk of the importance of an attribute to the patient. So clearly, they are different dimensions of importance. So often we look at bioactivity, pharmacokinetics, safety, and immunogenicity, and separately assess the risk for an attribute in all those areas. So this inverted triangle that goes from red to green, is a little bit of a simplification. One needs to stretch that because the truth is the vast majority of attributes we may just not know. And we may think they're low risk, but we may not have really clear data about that. And furthermore, there probably aren't the large subset of these that actually do matter to the patient. And that it may be that this is should be flipped, with the red being the lower part of this and the green being the bigger part of it. But the dilemma still is, how do you know which things really live in the red? And again, we use risk assessments. We have a lot of experience with products. I think, in general, we're fairly good at this, although occasionally we may be surprised. But that tells us which attributes are important. How do we know what the range of those attributes are? So there's a lot of information that goes into deciding on what those ranges are. Again, product and related product data, knowledge management, weighing risk. And so a very old way of looking at this, so on the y-axis is a readout, whether it's a bioassay or clinical studies or PK data or bioassays other than the validated bioassay. And then this might get looked at on only one lot, or it may get looked at on many lots. And usually what gets the most lots are the clinical study and the validated bioassay. And then we may have, data from some of these other assays on different lots, and all of that integrated into what the attributes are on those lots and what seems like a reasonable place to set specifications. Now, I include clinical studies here, but clinical data has a very, very limited role in supporting quality because exposing patients to intentionally variable material is not looked upon well, and clinical studies are one of the most expensive ways of answering a question. This was certainly imagined. So in an example of a quality by design case study from a number of years ago on a product called ACE, not to be confused with, ACE2 is now the receptor for coronavirus. This was well before that. But this imaginary product in the case study had a 6-arm PK study based on varying manufacturing things like, the lubricant levels, ribbon density, the API particle size, and looking at whether or not that impacts both dissolution and actual PK. So this was a hypothetical example, and I don't think any pharmaceutical company is likely to do a 6-arm study to define manufacturing. So how do you deal with this? And so I think, again, it goes to risk assessment and leveraging as much knowledge as you can. And most of that knowledge is biology. And in terms of where analytics are going, Barry Karger has said we should be looking at molecules, not attributes, right? Which is really the combinations of attributes in a particular molecule. And that, even in advisory committees for biosimilars, our peptide maps are averages. They're not really molecules. And so imagine actually data on specific molecules in the mix, a whole level of resolution. Again, with top-down and bottom-up methods. We talked about fingerprint-like, this could be one way of thinking about what that might mean. However, if this doesn't add value to the patient, it doesn't really mean anything. And so really the question is, how do we find out what matters to the patient and how do we set ranges? And it's about biology, whether it's the bioassays we're already doing, maybe the future when we get even more sophisticated. But biology and bioassays, I think, are going to be the driver on deciding on what level of characterization matters, where on this tree of sensitivity and resolution we need to be, and what attributes we need to focus on. So I want to flip this from this concept about biology being a key part of how we do quality to talk about a sort of different work stream, which is drug development in general. And using, this is old data now, but this is very public bio data on clinical development. So there are varying probabilities of success. Phase 1 to phase 2 is pretty good. You generally, once you're in phase 1, have a 60% chance of doing a phase 2 study. Phase 2 to 3 is the valley of failure. Basically, about a third of products make it from phase 2 into phase 3. Phase 3 to BLA, again, similar to an IND, making it to the next phase, about 60%. But once you're already in a BLA, you're close to 80%. Phase 1 to approved product is about 10%. But this 10% really varies. And if you look at non-NMEs, right? So the 10th statin or whatever, that when you're really not being groundbreaking, much higher rate, more than double, because there's a lot of risk that's removed by knowing that a very similar product works. Biologics tend to have a somewhat higher success rate. That may be because antibodies have a success rate, or there's a little bit more sense of the target and what's not a target for biologics. And then NMEs in general are actually well under that 10%. If you're a new molecular entity, it's a fairly low probability of success. And the causes of these failures, they can be safety, but efficacy dominated phase 2 and phase 3 failures. So more than half of those failures are not because of an unexpected safety issue. They're because the product simply didn't work, despite the research and development in moving it forward. Now, there are also some subsets. So, for instance, rare diseases have about a 25% chance of making it from phase 1 to approval versus all diseases. Products that have a biomarker involved also have about a 25% chance of making it from phase 1 to approval. Conversely, oncology drugs have a lower probability than other drugs of making it to approval. And there is some literature to suggest there's often such a rush to push oncology drugs forward that there's really not enough investigation into, is it a really meaningful target? So to me, all of this says, if you really have more data on the validity of your target, you are more likely to succeed, whether it's a biomarker associated with that target or a rare disease, which are often single gene defects where you really understand exactly what you need to do, that those are markers of drug development success. So target validation is a critical part of success. And there's literature about better use of new technologies like genome editing to better test whether markers are available. Drug mechanisms that have some genetic support have a much higher chance of being approved drugs. And so estimates that using genetically supported targets could double success. So how do you really tell whether your target is going to give you a higher chance of success? It's in silico, in vitro, and in vivo biological models. The same biological models that you might use to decide on a quality assay that's relevant should also be important for the drug succeeding in the clinical realm. And so once you know the target, what's the exposure of your product to the target relating to PK? Structure plays a role in that, and in localization. Do you bind? Obviously, structure and what happens to your product in vivo impacts binding, and then what happens at that level. Again, all of these things are things that are linked to product survival. So I am approaching 2:15. So this is the way I'm going to go through some of these things really quickly and head to the punchline. So we know of a variety of studies, public and some not public, where a single PK assay used mass spec to look for a variety of protein variants. An example here showing deamidation goes up rapidly in vivo, which might be very useful for deciding how important tracking deamidation is to begin with if it rapidly happens in vivo. PK studies have given us a lot of information about antibody PK. Many studies suggest high mannose matters, other structures may matter, but there are examples that we do not know in terms of PK. A number of examples in the literature where products clear rapidly and it's not clearly linked to glycosylation or neonatal FC receptor binding. And so there still remains areas where these matter, and also where do they localize? And again, there are a variety of imaging technologies that could be used. Often they're only used in something called theranostics, where you're interested in a diagnostic assay that will then inform later administration of another labeled or unlabeled product. But there's a lot of information that could be gained from these studies. And so it'd be really great if we could actually take a product, label it in a way where you not only know where it goes, but you know what subset of its structural forms are at that site. And so I just think analytics link to clinical performance through biology, and that biology matters for both of them. And one really needs to think about ways to leverage that. And I'm actually going to jump to the end. This slide basically takes, if you look at a lot of drug development swim lanes, many of them, whether they are early preclinical development, whether they are manufacturing, such as quality bioassays or determining CQAs, and whether they are decisions to invest in clinical trials for new indications or to develop bio-betters, all of those live in a hub where understanding the biology of the molecule is the center. And structure function is really the thing that links analytics to that. So I want to close with a concept about business model innovation. So there was a book and a number of articles in the Harvard Business Review about ways of innovating that are not about technology. They're about the business model you use. And they focus around questions like, what are your offering will be, or what will you be focused on? What is your business focused on? When you make decisions, do you make them early or late? Who controls the decisions, and why they are made? What are the drivers for those decisions? And with the idea that cutting edge biology is a key to a whole variety of things in drug development, not just quality, that the what should be focused on is an integrated hub of biology, analytics, structure, function, and pharmacology. That ideally, those shouldn't live in many, many different places, but in one place. Decisions should be front-loaded for information that comes from biology and analytics. It might be really useful to use those tools to avoid failed phase 2 studies, such as we've seen. And so understanding the importance of this information and using it early. I mean, many times forensics will say a product that failed, if you looked back, there were some signs that you didn't really understand the target well enough. Who should make those decisions? Clinical development may make those decisions in many cases, but if there is a biology hub that is expert in structure, function, analytics, and pharmacology, they should have some role in that decision. And the drivers for this, if the whole bioassay program is only paid for by quality and could be leveraged across the organization, there's a business case that it should be funded in a way that reflects value it adds across all the work streams, not just quality. So I have some examples about how FDA connects quality and analytics and both biosimilars and in research programs where our biology may impact both quality and PD markers. But basically, I wanted to stress on the idea of a shared framework for protein attributes that would enable connections across analytical methods, not just be a way of organizing spec, but way of wording analytics that drive control strategy, deep characterization, comparability, and similarity, and the idea of an integrated biology hub that is not only about informing quality, but should link to all of drug development and life cycles to create a tapestry that will really help us move forward with novel and effective medicines. So I am done, and I think I have a few minutes for questions. Thank you.
I'm looking forward to questions. If you have questions, please go into the chat line, and I will give those to Steve. Steve, I have a question for you. You were describing your database for molecules and getting all the pertinent information, characterization, et cetera. Do you have companies being reluctant that they're giving away maybe too much information to their competitors?
So I would say this. The whole idea of this is a framework is public, but the deep characterization data would not be public unless a company wanted to share that, right? And the companies are already sharing that with the FDA in particular applications. For us, it would just be helpful to have things organized in a reasonable way. And then for the parts of this that are worth sharing, it becomes very useful for industry. Even if a company never shares anything publicly, if the way that it organizes its information matches up to the way academics organize their information, they can better mine public information that is being shared. So this is in no way meant to impact what is proprietary or trade secrets. This is just saying whatever you share, which you're sharing anyway, that a common framework for that is of value.
Okay, I just remember, we've talked about this as a group in CAST for a couple of years, and I know that large pharma has always been a little reluctant to give too much out. So
Right, but this wouldn't force a company to do this, right? Or, again, ultimately there may be agreements in ICH about what data needs to get submitted, and that's very different than this. This is more about, is there a framework for which there's a preferred way to organize information? I mean, that exists in a lot of other ways. There are standards in clinical trials for how you describe adverse events, right? That doesn't mean a company needs to share all that. I mean, they do need to share that information as relevant and according to whatever the rules are, but that it's just a standard framework or a standard framework for how clinical data is submitted. It doesn't mean all of that needs to be shared. It means that there's an organized way of having it that is public. The data itself doesn't have to be public.
Okay. Julie, I'm not quite certain. I've got the 3 boxes up for questions, but I don't see anything coming through. So can you confirm I've got the right screen? I apologize. Okay, I've got a question here. Question for Steve. This is from Nadine Ritter. Can you update on the ongoing activities of the OPQ Center of Excellence in respect to OBP products?
So the OPQ Centers of Excellence are research-based. There are 5 centers of excellence. There's a pharmaceutical analysis one. There is a manufacturing one, which both has biomanufacturing and small molecule manufacturing. And then there are 3 biology-related ones, an immunology one, an infectious disease and inflammation one, and a tumor biology one. And those tend to be indications we see for biological products. Those COEs have investigators who publish their work. They also do other research-related activities. They might review BAAs that the agency puts out, and they may be consulted on specific, research-related or biological questions that a reviewer might have. And one of our goals in terms of this idea of saying biology is a shared resource is to really make that more available to the Office of New Drugs, which does clinical work and may be interested in a pharmacodynamic marker where the signal transduction pathway is the same as we've been looking at for a bioassay. And so to leverage that information. So, those COEs publish, based on investigator work, but they're also designed to sort of organize how we share information within CDER.
Okay, another question, Steve. Can you comment on the potential of the 4th dimension in your paradigm of resolution, sensitivity, and the relevance to include correlation between attributes? For example, the correlation of charge and higher order structure to safety, immunogenicity, and why efficacy/potency individually, maybe you follow a lower on the 3-dimensional plot, but correlate between attributes leading to higher relevance.
Right. So I think that touches upon the Barry Karger point about looking at molecules, not attributes. It may be the combinations of attributes are what matters. And I think that's actually very important to study. But if you could at least peg attributes in defined categories, you could then connect them. There'd at least be a defined way of expressing each individual attribute that then could be looked at that higher dimension. And I think that ultimately is going to be very important to look at and may matter for relevance in some cases.
Okay, another question, Steve. Do you use semi-solid media or liquid media for the FFA assay?
Right. So that's a very specific technical question. I don't think I'm going to address that. I think if you have a particular technical question related to a product, that it's best to work through your review team.
Another comment. They want to say congratulations on having 57 biosimilar products already approved in Morocco and 20 more in review. So guess maybe that wasn't for you, but is there any more questions for Steve at this point in time? Another comment is, Steve, inspiring presentation. The Biological Hub is a great way to inform drug development and decisions. Do you see knowledge gained through the hub to be used to justify wider specifications?
I do think that really good biological characterization can create more flexibility in defining the ranges of specifications. Again, it'll be a case-by-case review issue, and there may take some time for this concept to permeate, through regulatory agencies. But yes, because doing multiple clinical trial arms to define that simply is not practical. And the more we understand our molecules and the more comfortable we are with the relevance of the biology, I think the more flexibility we will have in creating quality expectations that match patient needs, which may mean they are broader in some cases than our manufacturing experience.
Okay, and then one more question. I know we're past the 5-minute mark that you promised you would give us. Can you comment on gene therapy products, AAV products, in terms of higher order structure?
So again, I'm part of CDER, so we don't really regulate gene therapy products. That might be better addressed to someone who deals with that area. Okay,
Steve, thank you very much. I very much appreciate on behalf of CAS your participation. Is very enlightening as usual. I'm sorry you had a moderator that's technologically challenged, but appreciate your time and effort. Looking forward to seeing you soon.
Okay, well, thank you, Bruce. You did a fine job despite your self-criticism. And thanks, everybody, for your attention.